Assalamu alaikum, everyone. Today is the first lecture for cell biology, the online version. And in the last class, we talked about the cytology and how they discovered all the different machines and adawat, like the electron microscope and so on, in order to learn about the cell structure. And now we continue with the biochemical strand. Okay, so the biochemical strand is looking at the different chemical reactions that occur in different parts of the cell. So as the cell biologists that we call cytologists, as they were studying, the structure of cells by using different types of microscopes. Well, at the beginning, they were very simple microscopes. Other scientists started to use, to explore the function of cells. So they were interested to see what is going on inside of the cells. And they were trying to really link the structure of the cell to the function of all the molecules, the macromolecules that are found inside of the cell. So the first scientists were Frederick Wöhler, and he actually showed that a compound that we call a biochemical, we could say, so a compound that is made in a living organism could also be produced in the lab. So what Frederick Wöhler did is that he mixed ammonium ion with cyanate and he produced urea. Now, urea is a biochemical that is found, as a waste product of nitrogen metabolism in our body. So this was, we could say, the first biological compound that was synthesized, artificially, we could say, in the lab. So something that appears in living cells can actually be produced by this chemical reaction in a laboratory. Now, a few years later then, in the 1860s, we had Louis Pasteur, who was a French scientist, and he was working on fermentation of sugar into alcohol. And he showed that yeasts are able to ferment sugar into alcohol. Okay, so this was like the first time that they linked, we could say, a chemical reaction with living cells. So the living cells were yeasts, which we know are eukaryotic, unicellular organisms. And these yeasts, if you put them in the presence of sugar, they produce a fermentation product, which was alcohol. Then in 1897, we had Edward Buchner, who's shown in this picture, and he showed that yeast extracts could also ferment sugar into alcohol. So what he did was that he broke up, he took the yeast cells and damaged the cell walls and membrane. So the juice of the yeasts, we could say, or the extracts, this could also do the same reaction. So they could ferment sugar into alcohol. You didn't need the living yeast cells. You could extract the juice, we could say, from the yeast, mix it with sugar, and this will produce alcohol. So this was really began the discovery of enzymes. And this word enzyme, the word zyme actually is Greek word that means yeast. So this shows us, that this was really the beginning of biochemistry, when these biochemists were now looking at how the enzymes, they catalyze biological reactions. So this was the discovery of the enzymes, in other words. So as the days went on in the 1920s and 30s, the scientists worked hard to determine all the different reactions of fermentation and other reactions as well. So these were explained quite a long time ago, in 1920s and 1930s. So we had Emden, Meyerhof, and Warburg, and Krebs. They did a lot of very important biochemical work. So we know, when the glucose is broken down in our cells to pyruvate, so 1 6-carbon compound is broken down to 2 3-carbon compounds, and this is called glycolysis. So glyco means sugar, lysis means break. So this was actually called the Emden-Meyerhof pathway of glycolysis. So they discovered the pathways of glycolysis. And of course, if there's oxygen present, then the pyruvate will normally enter the mitochondria. And in the mitochondria, we will have the Krebs cycle, where the pyruvate is completely oxidized to carbon dioxide and water. Okay, so all of this is important for producing energy. Now, energy can be produced by glycolysis. And if you remember from your biology classes, during glycolysis, we have substrate level phosphorylation, which produces a small amount of ATP. And then if there's oxygen present, the pyruvate that is produced by glycolysis will enter the mitochondria and be oxidized by the Krebs cycle. And here we also have a limited amount of ATP produced. And then, of course, we know now that the electrons are carried by the electron carriers, to the electron transport chain in the inner membrane of the mitochondria. And this is where the main bulk, the largest amount of ATP will be produced. So, as we said, these scientists started to look at the biochemical reactions. And both glycolysis and the Krebs cycle are important in producing energy by cells. Then the first person that actually showed that ATP, or adenosine triphosphate, is the main form of storage of energy in most cells was Fritz Lipmann. So he showed that ATP is the main source of energy storage, we could say, in our cells. Now, another important discovery in biochemistry, in order to study the different biochemical reactions, was the use of radioactive isotopes. So these include things like tritium, which is H3, carbon-14, P32, and S35. These are all radioactive compounds. So these compounds can actually be incorporated into biomolecules. That means we can radioactively label different molecules that cells produce. And this allowed the researchers to look at how these molecules are produced and how they move and how they get changed or transformed inside of the cells. Okay, so a very important, we could say, tool, was the use of radioactive isotopes to look at how biological molecules are made, how they move in the cell, and what happens to them, at the end. And in fact, I'm sure you've heard of the Calvin cycle, and the main discoverer, who is Melvin Calvin, he was the first person to use radioactive carbon-14, labeled CO2, that he incubated algal cells. These are the algae, Tahalib, and with a light source, they would do photosynthesis. So in the 1940s to 50s, the Calvin cycle was discovered, and this is the way that the most common way, we can say, for metabolism of carbon during photosynthesis. So therefore, the Calvin cycle was the first pathway of metabolism that was explained using radioactive compound, which was carbon-14. Another important discovery for biochemistry was subcellular fractionation. So you know that fraction means like 1/2, 1/4, Subcellular, we're talking about the stuff inside of the cells. So subcellular fractionation, therefore, is a technique that uses centrifugation, a centrifuge, to separate from each other different structures inside of the cell and different macromolecules. And by macromolecules, we mean all of those polymers like proteins and DNA and so on. So this is based on their size, their shape, and their density. So basically, if we have our cells and we homogenize them, we make like a cell shake by putting it in a Moulinex and breaking up the cell membrane, all the stuff inside of the cell will be present in this tube. And then this tube is taken and centrifuged at different speeds for different times. Now, the particles inside will come to the bottom of the tube according to their size, shape, and density. So this means, the largest structure inside of the cell, for example, is the nucleus. So after the first centrifugation, we will have the nuclei at the bottom of the cell. And then the liquid is taken, bottom of the tube. The liquid above the pellet, we call this, is taken and put in a new tube, and this is centrifuged at a higher speed for a longer time. And the next largest organelle, we could say, are the mitochondria, and if it's a plant cell, it will be the chloroplast. So step by step, the scientists were able to separate the different structures inside the cells, and then they were able to test them to see what type of enzymes they have, what type of chemical reactions happen. Now, in order to be able to isolate these different organelles, we can't use the normal centrifuges. you know, which don't spin very fast. So we have these super duper centrifuges that are called ultra centrifuge. So these are able to spin at very, very high speed. Okay, so over 100,000 rounds every minute, which is talking about more than 500,000 times the force of gravity, which is the jadabiyya. So in this way, you know, the scientists were able to isolate different parts of the cell and then maybe study their structure with the electron microscope. And the biochemists were able to study their function, you know, using different techniques to look at what chemical reactions are going on. Another set of biochemistry methods that were very useful was chromatography. So this is a technique that biochemists use to separate molecules from a solution, you know, so you have a solution and you have different molecules, and based on the size of those molecules, on their charge or their chemical affinity, there are different types of chromatography techniques that will allow them to separate. We will learn later on what we mean by this chemical affinity later on in the course. Another technique, I don't want to go into too much detail, is electrophoresis. So as the word says here, electro, this is talking about an electrical field. So an electrical field is used to move proteins, to move DNA or RNA through some sort of gel, you know, based on the size and the charge. And then we have another technique called mass spectrometry, which once we separate these, for example, proteins, we can use this mass spectrometry to know the size and the composition of the proteins. So this is showing us a very simple experiment. Maybe you did it in your Tawjihi classes or, you know, in your school biology. So here we have plant pigments, a mixture of them. This is put onto this kind of filter paper, we could say. And then there will be a solvent that this paper is put on. And as the solvent goes up the paper, it will separate the pigments from each other. And And over here, we see electrophoresis of protein samples. So this is a way that scientists can study proteins. They separate the proteins by electrophoresis. Then they can isolate these proteins and use mass spectrometry to determine their size and the composition of these proteins. And finally, we come to the genetic strand. Okay, so we talked about the cytology strand, which focuses on cell structure using optical techniques like the light microscope, electron microscope, the super resolution microscopes, fluorescence microscopy, confocal microscopy that we talked about in the class. The biochemical strand then is looking at the biochemical reactions, and a big discovery of biochemistry was the ultracentrifuge. And now we come to the genetic strand. So here we're looking at the flow of information from one generation to another. So we are studying inheritance, which is Wirathe. And here we have the father of genetics, Gregor Mendel, who was a monk in a monastery, and he did a lot of experiments in the garden of the monastery with his pea plants, which are the basilla. And his experiments in 1866 described how hereditary factors that we know now are genes, how they are transferred from the parental generation to the offspring. So the gene is the offspring. So these were very, very famous and important experiments in genetics. So Mendel is the father of genetics. Then around the 1880s, we had this scientist who was Walter Fleming. He was the first person to call chromosomes. So he saw these bodies in the nuclei that were like threads, like khiyut, and he dyed them with special stains, and those dyes were taken up by the chromosomes. So chromo means color, and zome comes from the word body. So he called them chromosomes. And then in a few years later, we had Wilhelm Roux, and quite quickly after Roux, we had August Weismann, who identified the genetic material is on chromosomes. So they said that the genetic material is found on the chromosomes. Okay, so this is repeated slide. Yeah, so when Fleming saw those thread-like bodies in the nucleus, he called them chromosomes, and he called the process of cell division mitosis. And then, as we said, Roux and Weismann suggested that chromosomes carry the genetic material. So this is Weismann. So when Mendel made his initial, when he talked about his results, not a lot of people were that interested. But as it happened, his work was rediscovered, we could say. So his initial results or findings were rediscovered in 1900s when we had these plant biologists that were working alone, each one. So they were working independently, and they all talked about Mendel's studies almost at the same time. Okay, so this was when the importance of Mendel's work was really realized. So we had Korinth in Germany, we had Ernst von Schermark in Austria, and Hugo de Vries in Holland. So these three scientists were working on plant genetics, and they were writing papers, and they all cited Mendel's studies at the same time. And within 3 years of this, the chromosome theory of heredity was made by the American doctor Walter Sutton and the German biologist Theodor Boveri. So these made this chromosome theory of heredity. And this is basically that heredity factors that we know are genes that were responsible for Mendel's inheritance patterns. These are actually found on the chromosome inside of the nucleus. So in this way, they identified that chromosomes carry the hereditary factors that was responsible for the Warathi of the bacilla in Mendel's experiments. And then this nadariya, or this hypothesis, was strongly confirmed, we could say, by Thomas Hunt Morgan and his students Calvin Bridges and Alfred Sturtevant. And these scientists were using, as a model, Drosophila melanogaster. So these are the fruit flies. And these scientists identified different types of mutants, morphological mutants of Drosophila, and they were actually able to link these specific traits that these flies had to specific chromosomes. So now we had the chromosome theory of heredity. Then people wanted to know what is really inherited. So the first person, who was Friedrich Miescher, he was the first person to actually isolate DNA, and he called it nuclein. And then by 1914, it was actually known to be a part of chromosomes. And they knew by the 1930s that it was made up of just 4 different nucleotides, which are the adenine, guanine, cytosine, and thymine. And the scientists couldn't believe that just 4 little nucleotides, are they really enough, to explain all the diversity that is seen in living organisms? What we mean by this is all the differences that we see in all the living organisms, from an amoeba to a sarsur, to a frog, to a mouse, to a human, okay? Just 4 nucleotides. They wondered how this could be enough to explain all the differences between these living organisms. Now, at the same time, they knew that proteins are made up of 20 different amino acids. So the scientists believed that the hereditary material was actually proteins, not DNA, because there are 20 proteins. So this would mean, that these could explain, these are enough to explain all the diversity, all the differences that are actually seen in amoeba and in the mouse and in the sarsur and in humans and so on. Now, scientists started trying to guess or trying to work out what is really the genetic material. So as you all know about this because you learned it in your biology class, we know that these different scientists did a lot of different experiments that showed DNA is the genetic material. So the most, we could say, one of the most important experiments that clearly showed that DNA is the genetic material was in 1944 when Avery, MacLeod, and McCarthy showed that DNA transformed the non-pathogenic strain of bacteria to the pathogenic one. So DNA was the transforming agent. And this DNA was inherited by the daughter cells. So if you remember Griffith's experiment, you know, when he was working with a non-pathogenic and pathogenic strain of Streptococcus pneumoniae, that's the experiment I'm talking about here. And they didn't, so Avery, MacLeod, and McCarthy showed that it's DNA that transformed the non-pathogenic strain to the pathogenic one. Then 8 years later, we had Hershey and Chase, and they actually showed that it is DNA and not protein that enters a bacterial cell when it is infected and altered by a bacterial virus. So you know that bacteria can be infected by viruses, just like we were infected with coronavirus. E. coli can be infected by a bacteriophage. So they did their experiments and their results showed that DNA is what enters the bacterial cell when it gets infected by the bacteriophage and when it gets genetically altered by the virus. And then we had Beadle and Tatum in the 1940s who were working with you know, the fungus that grows on the bread? This is called a mold. So the name of the mold was, you know, Fatriat Neurospora crassa. And Beadle and Tatum made the one gene, one enzyme concept. So they were the first ones to do that. And of course, then we went on with more important findings about the structure of the DNA. So in 1953, Watson and Crick, with great assistance from a female scientist, not a male scientist, from a female scientist, Rosalind Franklin, who was the first female, the first person in the world to take an actual photo, we could say, through her X-ray crystallography technique of the DNA molecule. So Watson and Crick analyzed her X-ray crystallography photos, and according to Rosalind Franklin's work, they proposed that the double helix, that the DNA molecule is made up of a double helix. And then, of course, as the time went by, in 1960s, we had a lot of different new information that helped to understand how DNA replicates, how RNA is produced, and about the genetic code. And Crick, he actually put together what is called the central dogma of molecular biology, which he said that the information in DNA has the ability to replicate itself, and the information will flow from DNA to RNA as the messenger RNA is transcribed from DNA. And then when the RNA is translated, the information will go to the protein. So this is the central dogma of molecular biology. The information flows from DNA to RNA to protein. And I'm sure that you've learned that there are three important kinds of RNA. We have the messenger RNAs, which carry the genetic message from the DNA, and they take it out of the nucleus through the nucleopore complexes into the cytosol to the ribosomes. And the ribosomes will be busy making proteins. Then we have rRNAs, which are ribosomal RNAs that are a part of the structure of the ribosomes. And we have tRNAs, T for transfer. These are in between, intermediaries, al-wasata, something like that, that will recognize the coded base sequence of the messenger RNA, and they will bring the correct amino acids to the ribosome so that the ribosome can join them together in order to make a protein. Now we have to remember that normally, the genetic message goes from DNA to RNA to protein. But there are exceptions to this rule. And this includes viruses that have not DNA as their genome, but RNA genomes. So these will synthesize messenger RNA using RNA, not DNA, as the qalib or as the template. And other RNA viruses like HIV have the process of reverse transcription, where the viral RNA is actually used as a template to make DNA. So here, there is a backward flow of the information, the genetic information, from RNA to DNA. So the main idea from this slide is that, normally the information goes from DNA to RNA to protein. But with some viruses, we have a different story. We have exceptions, meaning they don't follow this rule. And as we said, that some viruses that have RNA genomes, they make a messenger RNA from the RNA. Other viruses like HIV, they have enzymes, special enzymes that do reverse transcription, which means that from the RNA, they make DNA. Okay, so this is the flow of genetic information. We have the information in the DNA molecule, and this information will go into messenger RNA. So the nuclear DNA will give information for the synthesis of messenger RNA, and then this comes out of the nucleopores. They will go to the cytosol, and there they will bind to the cytoplasmic ribosomes, and we will then have translation of the messenger RNA, the message in messenger RNA, where the message will give information to the tRNA on which amino acids to bring, and these little amino acids are transferred, and then we see a polypeptide starting to grow from the ribosome. Now, Recombinant DNA technology started since the 1970s, and this allowed us to know a lot about gene expression. So in this technique, the scientists can use enzymes that are called restriction enzymes, and these enzymes can cut the DNA at specific positions, and this will allow scientists to produce recombinant DNA molecules that can be made with DNA from different sources. So this kind of technology led to DNA cloning, which is generation of many copies of this particular DNA sequence. And alongside here, we can also have a process of DNA transformation. So this is when scientists can actually introduce DNA into cells. And we know that the scientists developed DNA sequencing methods very, very quickly to determine, the base sequences of the DNA molecules. And nowadays, this is applied not just to a single gene, but, to whole genomes. And in the olden days, it took so long, to do this. But now we can actually sequence entire genomes. So this is like all the DNA in a cell, which is like 3.2 billion bases in humans very, very quickly. And then we have the development of bioinformatics, which is basically bringing together computer science with biology to organize and to understand a lot of sequencing data. So all of the sequencing of different genes and genomes, it has to be handled somehow, and this is too much for a human. So now computer science is merged with biology in order to, first of all, organize, and secondly, to understand all the sequencing data that we get. So in the human genome, for example, there are 20,000 protein coding genes, and half of these were not actually recognized before this genome sequencing. So nowadays as well, we have these science of omics. So genomics is studying the genes, all the genes of an organism. Proteomics is studying all the functions and all the interactions of every protein that is present in a cell. So we could call the genes the genome and the proteins the proteome. And another important thing is that scientists have been working on model organisms, and these have been very important in modern cell biology research. So we have a number of different model systems that allows us to look at what goes on inside of cells directly in living cells and organisms. Now, cell cultures are also a model system. So these cell cultures are normally taken from cells from our body, and they're used and grown in the lab in tissue culture. So the cells themselves can be used as a model system. But when we're talking about model organisms, we can say that most of what we know about a lot of different cell processes was actually used, learned using cells of E. coli, which is a bacterium, as a model system. And for eukaryotic cell cycle and protein interactions, scientists use a yeast, which is a eukaryotic organism, Saccharomyces cerevisiae. So this is used to study the cell cycle and protein interactions. We also have the wild type Drosophila melanogaster, which is the fruit fly. So this is a very good system to use because this is a multicellular organism. It's a eukaryote, and it has a short generation time, so this makes it very good. And this is used, for example, to study embryogenesis, how the embryos develop. So it's important in science of developmental biology, as well as cell signaling, how cells talk to each other. In order to study these processes, we need multicellular organism. So this Drosophila is a multicellular organism, easy to grow, short generation time. So we can have hundreds and thousands of flies very, very quickly. And in this picture, we see C. elegans, which is a roundworm. And as we see from this microscope photo, that is quite transparent, شفافي. So we can actually see the cells. We can see individual cells. And if we inject the worm with fluorescently labeled antibodies, for example, to stain a particular structure, we can actually see the fluorescently labeled molecules inside of the organism. And this is used to study cell differentiation and development. So it's a very useful organism, model organism. And of course, we have the mouse, which is called mus muscula, which is the house mouse. So this is very good because this is a mammal. So it's different, we could say physiology, its normal processes are very similar to humans. So these are used often in biomedical research, if we want to study different types of human diseases like osteoporosis or any kind of disease, cancer, and so on. Infection, microbial infections, they can inject the mice and see what happens and where the bacteria or viruses, what kind of changes they can make. So this is an important model organism. And finally, we have Arabidopsis thaliana, which is, the khardal or mustard plant. So this is very useful for doing plant biology research. Okay, so that's all that we have for today. Now, remember that your lecture will be in PH2 102 from now on. So it has been moved from the engineering to the PH. Okay, so please check before you go to see which room it is. So that's it for today, and I will see you next week, inshallah. And I will end the meeting and put this recording on the e-learning. Bye.